There is a process happening inside you right now that began before you took your first breath and will continue without interruption until the last moment of your life. It is so constant, so seamlessly woven into the fabric of your existence that most people never think about it at all. But here is something that changes the way you understand it. The digestive system is not simply a tube that processes food. It is, in every meaningful biological sense, an autonomous world. One with its own nervous system, its own immune forces, its own chemical language, and its own sense of time. What we call digestion is not a single process. It is a carefully orchestrated cascade of events involving dozens of organs, hundreds of enzymes, trillions of microorganisms, and electrical signals traveling faster than conscious thought. And it begins, as most profound things do, long before you are even aware it has started. The moment you see food, not taste it, not smell it, but merely see it, your brain fires a signal down the vagus nerve, one of the longest and most complex nerves in the human body. Your salivary glands activate. Your stomach begins producing acid. Your pancreas releases a small advance wave of digestive enzymes. The body, in a very real sense, starts eating before a single bite has been taken. This anticipatory response, known as the cephalic phase of digestion, is not a curiosity. It accounts for roughly 20 to 30% of the total digestive capacity your body will use during a meal. The sight, smell, and even the thought of food primes an entire biological infrastructure for work. This is why eating while distracted, while staring at a screen, while under stress, while rushing, genuinely and measurably impairs digestion. The opening signal has been missed. Once food enters the mouth, the first transformation begins. Chewing is not simply mechanical breakdown. The act of grinding food activates the release of salivary amylase, an enzyme that begins cleaving the chemical bonds within starch molecules on contact. Amylase does not simply coat the food. It begins dismantling it at the molecular level, breaking long chains of complex carbohydrates into shorter fragments called maltose. Simultaneously, the tongue and soft palate are performing an extraordinarily precise act of discrimination, evaluating temperature, texture, moisture, and chemical composition, relaying that information to the brain in real time. There is a reason some foods need to be chewed many more times than others. Thorough mechanical breakdown dramatically increases the surface area available to digestive enzymes later in the process. A principle that, if ignored, creates a cascade of inefficiencies further along the tract. The bolus, the compressed, moistened ball of chewed food, is swallowed and begins its descent through the esophagus. What happens here defies the simplicity its name suggests. The esophagus is not a passive pipe. It is a muscular structure lined with specialized tissue that contracts in coordinated waves, a mechanism called peristalsis, pushing the bolus downward with rhythmic precision. These contractions are so powerful and so well organized that you could swallow food while standing on your head, and peristalsis would still move it toward the stomach. This is not merely interesting anatomy. It reveals something important. The entire digestive tract is lined with a network of neurons called the enteric nervous system, sometimes described as the second brain. This network contains approximately 500 million neurons, more than the spinal cord, and can operate entirely independently of the brain in your skull. It monitors the contents of the gut, adjusts movement, regulates secretions, and communicates constantly with the immune system and the central nervous system. Long before neuroscience began mapping this system in detail, people had intuited its existence through the sensation most of us recognize as a gut feeling. At the base of the esophagus sits the lower esophageal sphincter, a ring of muscle that, under normal circumstances, remains tightly closed. When the bolus arrives, a carefully timed signal causes it to relax and open. The food enters the stomach, and the sphincter closes again. What waits inside the stomach is not what many people imagine. It is not a bag of acid that dissolves food by brute force. The stomach is a muscular chamber capable of generating extraordinary mechanical force through churning contractions, and it also secretes a highly controlled mixture of hydrochloric acid and proteolytic enzymes, most notably pepsin, that begin dismantling the protein structures in food. The acidity of the stomach contents at peak secretion, with a pH that can drop to around 2, is comparable to industrial acid. Yet the stomach does not digest itself. This is perhaps the body's most underappreciated engineering achievement. The stomach lining is protected by a thick layer of alkaline mucus, constantly renewed by specialized cells that neutralizes the acid on contact with the tissue surface. The moment something disrupts that mucus layer, chronic stress, certain medications, the bacterium Helicobacter pylori, the acid reaches the tissue, and the result is an ulcer. Not a deficiency of acid control, but a failure of the protection system. After 30 minutes to several hours of churning, depending on the composition of the meal, the stomach has converted its contents into a semi-liquid substance called chyme. This is no longer food as it was consumed. It is a chemically transformed mixture of partially digested proteins, fragmented fats, broken down carbohydrates, and acid, ready for the most complex phase of the digestive process. The pyloric sphincter, another muscular gate, this one at the base of the stomach, begins releasing chyme in small, precisely timed pulses into the first section of the small intestine, the duodenum. The timing matters enormously. The small intestine is not designed to receive a flood of acid-rich material. It receives it in controlled installments, and it responds to each installment with a coordinated chemical reply. As chyme enters the duodenum, it triggers the release of 2 hormones, secretin and cholecystokinin, from specialized cells in the intestinal lining. Secretin travels through the bloodstream to the pancreas, signaling it to release a bicarbonate-rich fluid that neutralizes the acid arriving from the stomach. Cholecystokinin signals both the pancreas and the gallbladder simultaneously. The pancreas responds by releasing a cocktail of digestive enzymes: lipase to break down fats, proteases to continue protein digestion, and additional amylase for carbohydrates. The gallbladder contracts and releases bile, a substance produced by the liver and stored in concentrated form, into the duodenum. Bile does not digest fat in the chemical sense. It emulsifies it, breaking large fat globules into microscopic droplets, vastly increasing the surface area available for lipase to act upon. This process, invisible to the naked eye, is the reason dietary fat can be absorbed by the body at all. Without bile, fat passes through the intestinal tract largely unprocessed. The small intestine is where the majority of nutrient absorption takes place, and its architecture is designed around a single physical principle: the maximization of surface area. The inner lining of the small intestine is not smooth. It is folded into large circular folds called plicae circulares, and each fold is covered in finger-like projections called villi. Each villus, in turn, is covered in even smaller projections called microvilli, forming a dense brush-like surface known as the brush border. When all of these folds and projections are taken into account, the absorptive surface of the small intestine, a tube roughly 20 to 23 feet long, is estimated to be around 2,150 to 3,200 square feet. That is approximately the surface area of a tennis court, folded into a space that fits comfortably within the abdomen. The body does not simply create space for absorption. It engineers it. This surface is not passive. The cells of the brush border, called enterocytes, are among the most metabolically active cells in the human body. They express hundreds of specialized transporter proteins on their surfaces, each designed to capture specific molecular fragments: glucose, amino acids, fatty acids, vitamins, minerals, and carry them across the cell membrane and into the bloodstream or lymphatic system. Glucose and amino acids pass into the capillaries of the portal vein and are carried directly to the liver for processing. fatty acids and fat-soluble vitamins reassembled into structures called chylomicrons within the enterocytes, pass instead into the lymphatic vessels of the villi, and eventually reach the bloodstream via the thoracic duct. This dual-channel system, blood for water-soluble nutrients, lymph for fat-soluble ones, reflects an elegance that took centuries of anatomy and biochemistry to fully decode. Throughout this process, something else is happening that does not appear in most textbook descriptions of digestion. The gut is under constant immunological surveillance. The intestinal lining is home to the largest collection of immune cells in the human body, sometimes described as gut-associated lymphoid tissue, or GALT. These immune cells face a problem that has no parallel elsewhere in the body. They must distinguish between harmful pathogens and the trillions of beneficial microorganisms that colonize the gut, while simultaneously tolerating the molecular fragments of food that are chemically similar to both. This discrimination is not perfect. When it fails, when immune cells begin reacting to food proteins as if they were threats, or when the balance of gut microbiota is disrupted, the result can range from inflammation and food intolerance to conditions as severe as Crohn's disease or ulcerative colitis. Research into the gut immune system continues to be one of the most active frontiers in medicine, with evidence suggesting that disturbances in gut immunity may play a role in conditions as seemingly unrelated as autoimmune disorders, metabolic disease, and even aspects of mental health. The gut microbiome deserves its own moment of consideration. The large intestine, the colon, receives material from the small intestine that the body cannot digest on its own, primarily dietary fiber, resistant starches, and complex plant compounds. This material would be largely useless without the microbial community that inhabits the colon. Hundreds of species of bacteria, archaea, fungi, and viruses ferment these substrates, producing short-chain fatty acids, butyrate, propionate, and acetate, that serve as the primary fuel source for the cells lining the colon and contribute to the maintenance of the intestinal barrier. Butyrate in particular has attracted significant research interest, with studies indicating its role in maintaining colonic health and evidence suggesting it may influence gene expression in ways that affect inflammation and cell survival. The microbiome is not a passenger in digestion. It is an active participant whose composition varies between individuals, shifts with diet, age, medication, and environment, and whose disruption, called dysbiosis, is increasingly associated with a range of diseases that science is still working to fully understand. Water, electrolytes, and any remaining absorbable nutrients are recovered from the intestinal contents as they move through the colon. The colon absorbs roughly about 1.5 quarts of water per day from this material, gradually transforming liquid chyme into solid waste. The muscular contractions of the colon, slower and less frequent than those of the small intestine, move the contents forward over a period of 12 to 48 hours, depending on the individual and the composition of what was consumed. Several times a day, a stronger coordinated contraction called a mass movement pushes contents closer to the rectum. The rectum itself does not simply wait passively. It contains stretch receptors that signal to the brain when it has become sufficiently full, initiating the conscious sensation of the need to defecate. A moment in which the enteric nervous system and the central nervous system negotiate in real time, balancing physiological pressure against social context. Even this final act of the digestive process is a collaboration between the body's autonomous network and conscious control. From the first anticipatory signal in the brain at the sight of food to the final muscular coordination of elimination, the digestive system operates as a system of almost incomprehensible sophistication, one that processes roughly 30 tons of food over the course of an average human lifetime, extracts the molecular building blocks of every cell, tissue, organ, and thought, and does so with a level of chemical precision that no industrial process has come close to replicating. It adapts to seasonal changes in diet, compensates for illness and injury, communicates with the brain through hormones and neural signals, coordinates with the immune system, and maintains its own internal population of microorganisms in a balance that took millions of years of coevolution to establish. In this sense, digestion is not simply what the body does to food. It is what the body does to survive, to think, to move, and to persist. What makes this system most remarkable may not be its complexity, but its continuity. It does not pause when you sleep. It does not wait for a convenient moment. From the first meal of infancy to the last of old age, it maintains its rhythm without rest, breaking down the external world and rebuilding it into the substance of a human life. In every ounce of food absorbed, in every enzyme released, in every microbial fermentation happening in the dark interior of the colon, there is the quiet evidence of a design so refined by evolutionary time that to call it simply digestion is, in every meaningful sense, to understate what the body has become. If this journey inside your own body changed the way you see something as simple as eating, imagine everything else you've yet to discover. Subscribe to Anatomy Unlocked and join us in every episode as we uncover the systems that keep you alive without you even noticing.